starting today. He'll be giving another talk as well. He's done a lot of his actual work that's perhaps most prominent, some fascinating stuff that he's done with James Setna, with information geometry and the foundations of statistical inference, I guess one will say. He also does a lot of work with biology and with at least non-equilibrium statistical physics. He's a, so to speak, purebred Ivy boy, Brown to Cornell to Princeton, and now he's a professor in physics at Yale. So, he hasn't been in any decent institutions, but well, nonetheless. And if you're curious, yes, he is affiliated, shall we say, with John Machta, external faculty. Both of them are now visiting here regularly, and both of them are actually contributing a hell of a lot to the community. So as I think I may have mentioned, he'll be giving 2 talks, and this is the first. Take it away, Ben.
Thank you very much.
Let's see.
Yeah, thank you very much, David. It's a wonderful visit, a wonderful time to spend my Omicron. So this is actually the first in-person talk I've given in 2 years. So this is very exciting for me. Yeah, so I will get into it. There we go. Okay, so I want you to just take this all in. Don't look too carefully. What I want you to take from this is that we really know an enormous amount about biology's molecular details. So all the circles there represent proteins. In many cases, we know their structure. The arrows represent interactions, and those are things that we really know about the system. Some of them are phosphorylation reactions, some of them stick to each other. And these are all the things that are going on inside of a T cell. I'm not going to be talking about T cells today, but maybe they're on everybody's mind a little bit. And for an example that I am going to be going into detail about, here is the key components of a chemotaxis pathway in E. coli. I'm going to go into more detail about all these players, but those interactions are very well mapped and understood. And in particular, you can even read out the phosphorylation state of that protein marked Y. You can do this in real time. I'm going to tell you a little bit about how that works. And you get remarkable single cell traces like this. So that's what we know about the microscopic details. Then
no, you said we get beautiful single cell information, and then what's y-axis? What does it mean?
Yeah, so I will spend more time talking about this, but very roughly, that protein marked with a Y gets phosphorylated, so it gets chemically modified in real time. And you can track how much of it is phosphorylated as a function of time using a very fancy fluorescence tool. And this is what the traces look like. So you can see what, in this case, that those 5 bacteriums there are thinking about their environment.
This translates to whether it's running or tumbling. Is that what?
Yes. Yeah. So this is going to be the signal that controls run and tumble. Okay. So that's just a very brief survey of how much we know about the microscopics. We also know a lot about higher level functions. So there's a T cell that's attacking what it's decided is a cancerous cell. This is an SEM image. And then to go back to E. coli, this is images of individual bacteriums undergoing this run and tumble navigation that I'm going to tell you about. So you can see they swim in straight lines, and then they tumble and choose a new direction. Okay, and what I want you to think about is how do you quantitatively connect these? And how do you connect this sort of enormous amount of detail that we know at the microscopic level in a quantitative way to these macroscopic behaviors? And I'm going to advocate for a reverse engineering approach. And the idea is that you want a simple but quantitative model of the behavior. And what you want from this is to be able to identify what it is that makes this behavior difficult for the organism to accomplish, and what the needs or the system specifications for the molecular details that are going to carry it out. And then have quantitative models and measurements at the microscopic scale that can try and identify how it is that these are met. And the goal is really to try and understand the design principles and how it is that biology does these macroscopic behaviors through its molecular details. Okay, so that's the big framework, but I'm going to tell you about two specific projects. The first is using information theory to connect chemotactic performance in E. coli really to the fidelity of their signaling pathway. First, I'm going to tell you that a fixed information rate provides an upper bound on how fast E. coli can chemotax. And then I'm going to talk about some experiments where we can identify how much information they get and compare that to the bound. And then the second part of the talk, I'm going to talk about quantifying the energetic costs of sending information. Here's one example that I'm going to spend a lot of time on. So a lot of signals are sent by electrically by depolarizing the membrane. And I'm going to talk about how much energy this requires and when biology should use different strategies. Okay. So I'm going to start in with this first part of the talk and tell you about chemotaxis and how it's information limited. And I just want to start by telling you about my collaborators. So this is a collaboration with Thierry's group that I really started to get interested in through talking with Henry Mattingly, who I knew when he was a grad student and I was a postdoc. Now he's doing a postdoc with Thierry, and Keita has developed this really spectacular Fred assay that I'll tell you about. Okay, so this is really about sensory motor control. And to get you thinking about it, what's going on in this nature scene here, these animals are taking in information from their environment and they're using it to control their muscles. And there's really two things that are going on here. So first, they're getting their sensory inputs, and that's controlling their motor behavior. But there's also motor feedback. So this is going to make quantifying the information somewhat difficult. So for example, if the cheetah there kicks its leg, that may change what its eye sees, but that's not really motor control. And so we're going to have to address this. And the very specific question that we're going to be asking is, do organisms efficiently use the information they're getting to control their behavior? So are they using all of the information that they get or using it in some sort of optimal way, or are they getting a lot of information and then not using it well? Okay, so I want to make an analogy to image compression. So as you can see, maybe depending on your internet connection, they all look bad. But what you should see is that as you go from A to D, these images get clearer. And one thing you could do is ask how much information you need to store these different images. And one way to frame this it is with a curve that separates this into a forbidden and allowed region. So if I plot image quality on the y-axis and image size in bits on the x-axis, there's an allowed region. So I can, yeah.
I don't mean to derail you, but don't you have to define quality?
Absolutely. I'm not going to for this example.
Right.
People think a lot about that here. So the y-axis is not an information theory quantity.
Got it.
That's something that you need to think about. Our visual system, perhaps. Yeah. So actually, this is harder to do for this example than for chemotaxis. Yeah.
And so by forbidden, you just mean that green curve is an upper bound?
Yeah. So if I want an image of this quality that takes this many bits, I can do that. If I want to make it higher quality, I can't do that. Okay. And just
is the shape of this curve something important, or is it just?
This is just a sketch. It will be calculated when I get to the actual example. But yes. Yes. Okay, and just to stress this, this is also possible, right? So in fact, if you save a poor quality image in PowerPoint, you often run into this. I suspect that's what's happening here, but I don't know. So, so you don't need to be efficient, but you could be. And the question I'm going to be asking is whether organisms, if we can make a curve like this and then see where organisms sit on it. Okay, so this is embarrassingly enough, I thought this was a microscopy image, but this is an animation, which I still think is nice, of the behavior that I'm going to be talking about, run and tumble navigation. These animated bacterium are trying to go towards the sugar, and the way they do this is they swim, and they alternate swimming with tumbling. Here's the real thing here. Maybe you can see if you follow the guy coming in from the upper left. It's more you can see he runs in a straight line for a while, then tumbles and chooses a new direction. The reason that we're going to start with this is that this is really the best understood behavior in all of biology, I would claim. It's very simple. We know exactly what they're trying to do. And we really know many of the components that carry it out. And that's what I'm going to be telling you about. But to give an overview of all the things I'm going to say here, the first thing I'm going to claim is that we can draw a curve like that image curve that I showed before, but now where the drift velocity, so how quickly these bacterium can climb a shallow gradient, is bounded by their information rate with some additional factors. Then I'm going to tell you about our measurements to actually calculate the information rate in these bacterium. And then I'm going to ask the question of how close actual bacterium are to this bound. Okay, so starting with this, I'm going to tell you a little bit more about the details of how they carry out this behavior called run and tumble navigation. So they have two forms of motility. One is this straight swimming run that you see there. And the other thing that they can do is tumble. And the same motor does this when it runs in reverse. These filaments splay out and they choose a random new direction, more or less. And I should tell you why they do this. So one of their main constraints is that they're very small. So E. coli are about 2 microns long, and the gradients that they'd like to climb are more in the centimeter range. So one thing that you could imagine they do is measure the concentration difference between their two sides and swim towards the one that's more favorable. And you can sort of work out the math for this, and it doesn't seem promising, right? That there's just not going to be a big difference between the two sides. Another major constraint for them is rotational diffusion. So they're small enough that thermal fluctuations will kick them in a random direction. And it turns out they can only go in a straight line for a few seconds at a time. So the strategy that they use to climb gradients is that they run in a straight line, or as straight a line as they can. And as they do this, they continuously measure the concentration. And if they think they're going uphill or towards attractants or away from repellents, they keep going. And if they think they're going downhill, they tumble, they choose a new direction, and they swim in that direction. Okay, and just very qualitatively, this is an example of sensory motor coupling, right? They're taking in information from their environment, they're using it to control the motor, and you can convince yourself that with no information, they can't climb a gradient, right?
Ben?
Yeah.
So there's an even simpler strategy if there's some set point of high or low sugar concentration, which just says if it's high, stay where you are, and if it's low, go somewhere else. Which yeah. I don't know if that's functionally different from measuring the time derivative, because presumably you would have to change the sort of set point based on your current environment. But I'm just curious whether, do we know that it's really measuring derivatives as opposed to just levels, to the extent that's a well-defined question?
Yes, it's very clear that they're measuring a derivative here. I'll show you the experiments. So maybe hold this question, but one of the really striking things about this system is that they can measure. So you can plug in how fast they go in a 10 millimeter gradient, for example, and they'll see about a 1% change in a typical run. So they do need to measure a really tiny concentration difference. And this system will work over several orders of magnitude of background concentration. So in a certain sense, they really don't care at all about the total concentration. They only care about the rate of change.
Thanks.
Okay, so maybe to answer this previous question, what the y-axis is going to be for this talk is drift speed, right? And that's the thing that's like image quality, but is much more easy to carefully define. That's what they're trying to do. That's going to be our performance measure. And the information rate that we're going to use is how much the passive S of t influences the current decision of whether to tumble or not. Okay, so what information does a chemotaxis bacterium need? And I'm going to take you through how we came to what we got. The first thing we wanted to do is the mutual information rate between the signal they're seeing. And I should say the signal is the rate of change of concentration. And I think another thing that I didn't say is we're always going to be considering just a flat gradient.
I'm sorry, I've already lost all the symbols. What is S of t? What?
Yeah. Sorry. So S of t is the instantaneous rate of change of the concentration. So that's what I'm calling the signal. And M is the state of the motor. So the motor can either be running or tumbling. So the naive thing that we started with was the mutual information between the signal and the motor state. And I think actually the easiest way to explain why this doesn't work is here. So it turns out in this simulation, there's no sensory motor coupling. So they're just running for a while, they take a tumble, and then they run for a while and they take another tumble. But there's a lot of mutual information between these. So if I give you this curve, you can tell me exactly where the tumbles happen, because that's when that's when there are these sharp spikes, right? That's when the bacterium chooses a new direction. And so the mutual information does contain sensory motor coupling, but it also contains this feedback from the motor system back onto the sensory system.
Sorry, I'm going to ask a really naive question. Can you define mutual information? Yes.
So the mutual information is if I give you two stochastic variables, it's if I tell you the trajectory of one variable, how much does that reduce the entropy of the other variable?
It's an information theory, generalization of covariance.
Okay.
So
Yeah.
defined in terms of entropies rather than square differences. So it's a couple of symbolic dynamics.
I could write an equation or two on.
I don't want to derail you now.
Yeah, I guess it's this was a good explanation. So if I have two trajectories, the mutual information between them is exactly how much the entropy of one of them is reduced if I tell you the trajectory of the other. And I guess from that definition, if I gave you this trajectory here, that reduces the entropy of the motor trajectory, right? Because whenever there are these spikes here, that's when the motor tumbled, right? Okay, so this mutual information can be larger than zero, even if I don't have receptors, right? So even if I'm not connecting the sensory system to the motor system. And so somehow we want just this part here. And so instead we use what's called the transfer entropy rate from S to M. And maybe I won't go into details about this, but the total mutual information rate can be decomposed into a sum of transfer entropies in these two directions. And we just want one of these.
Can I ask? So
Yeah.
somebody from the control theory world would say this is a Bayes optimal control problem. I would have a prior distribution over possible gradients of concentration, and I would, depending on my discounting rate into the future, I would come up with a policy that would actually have nothing to do with information theory, but that would be, in a Bayesian sense, the Bayes optimal control policy. So why should I be thinking about this in terms of information theory in the first place?
Yeah, so a different way you could ask, or you could get at something similar, is saying, you know, if for example you're counting molecules, people have thought about, you know, given the molecules that you've seen and their time trajectory, should you tumble or not? What we want to do differently here is we want to understand how the sensory apparatus limits how well you can do this, if that makes sense. So presumably the sensory apparatus can't literally count molecules. And
that would fall into a standard control theory, a PID control or something like that. Basically, I mean, all sensors in the real world are noisy. The airplane that you flew to come out here was based upon control theory, where you assume some noise in the sensors and you have models of the noise.
Yeah.
And so on and so forth. So I'm not quite sure why a priori I would expect an information theoretic approach rather than a Bayesian decision theory approach to be the right one to use.
Yeah, I don't quite see how you would get the,
I would have to define my objective function, which would be this kind of pure amount of whatever the particular chemical is that the E. coli is trying to accumulate is.
yeah, I think the objective function, you could use the same one that we're using, which is just you want to climb the gradient as quickly as possible.
Okay.
I guess what we're really after is the question of is the limit on how well they can climb chemotaxis that their sensory apparatus isn't better. So I don't swear that there wouldn't be a way to address this with a Bayesian approach. Yeah. I don't quite see how it would all go through, but it's possible. Okay. So to go a little bit further with this, I need to tell you a little bit more about these behavioral parameters. And what I mean by behavioral parameters are these are parameters that are well defined even if there's no sensory apparatus or no gradient sensing. Experimentally, if you put these bacterium in a solution that's just uniform. There are these 5 parameters. So this is the rate at which the bacterium tumbles. This is the average rate, sorry. This is the average rate at which you exit a run and the average rate at which you exit a tumble. Alpha is kind of the strength of a typical tumble. So literally the cosine of the angle between the starting and initial directions. dr is a rotational diffusion rate, and v naught is the run speed. So these you can measure for a bacterium that's not in a gradient. They don't require any information. And then in our model, the only way that the signal enters is that the rate at which runs are exited. So the tumble rate is going to be some arbitrary functional of the past of the signal, if that makes sense. So this can be some arbitrary functional of the past of the signal. And then we're going to define I dot to be the reduction in entropy of tumble decisions, given that you know the past of S of t. If this makes sense. So this transfer entropy rate, what we're going to be using is the reduction in entropy of whether you make a tumble or not, depending on the past history of signal. Okay, so once I specify all these parameters, that gives a drift velocity and an information rate. And then I can just optimize the drift velocity over these functionals, and this gives a bound on how quickly they can climb a gradient as a function of their information rate. And this is what it looks like. So there's this prefactor f of theta, which depends on these behavioral parameters. And then the drift velocity is bounded by this log 2 I dot over 12 dr. This is the rotational diffusion constant to the 1/2 power. I will talk about these experiments a little bit more, but this function is bounded between 0 and 1. So the best you can do if you choose your behavioral parameters is to make this 1. And it turns out from experiment, this number is about 0.5. If I can explain why that is, the optimal strategy is to set the tumble rate, so the rate at which you exit a run into a tumble, to be equal to the rotational diffusion constant. So you want to tumble about as much as you lose, or about as often as you lose your heading from rotational diffusion anyway. And it turns out that these cells tumble way too much. That's a little bit of a mystery. We and others have some ideas. So you can argue for them tumbling too much without these particular methods. But maybe we won't go there for now. Okay, so by measuring these behavioral parameters, we really get this curve, right? And that lets us make a plot like this. So on the y-axis now, there's the drift velocity divided by the run velocity. And on the x-axis, there's the information rate in bits. And just to give one sort of mini conclusion already, there's sort of a naive expectation that climbing a gradient in this way, you should get about 1 bit per run, right? So every time you run, you should get some information about whether you're going up the gradient or down the gradient. But actually, with 1/100th of a bit per second, and their runs are about a second, they can already climb gradients at 5% of their run speed, which is about how they do in a long gradient. Okay, so far I've been arguing that the information rate, the information that comes into their sensors, places a bound on how well they can chemotax. And what I want to spend the rest of this part of the talk doing is placing E. coli on this map. So where do they sit on this? Are they like these well-compressed images that they're close to this bound, or are they very far from it? So they're getting lots of information, but not chemotaxing well. Okay. So now I'm going to tell you a little bit of a detour into what we know about how this pathway works. At the front of the cell, there's a sensory apparatus that's composed of a bunch of receptors. And the receptors for attractants inhibit an enzyme, a chemical kinase, that's called key A here. And when key A is not inhibited, it phosphorylates this molecule key Y. And key Y diffuses to the motors. And when it binds to the motors, it tells the motors that they should run in reverse, and that initiates a tumble. So in that sense, when there's not enough attractant around, or when there's a repellent, this pathway sends a signal to the motors that tells them that they should initiate a tumble. Okay, so there's really 2 key features of the system, and you can see them here. So in this experiment, attractant is applied at this blue arrow here. There's this readout I'll tell you a little bit more about in a second, But you can think of this as measuring what fraction of QI is phosphorylated. And what you can see is when attractant is applied, the signal drops because the cells interpret this as they've just, swam into something good. They should keep going in that direction. But if you wait for long enough, the signal goes back up. So, they've been there for a while now and it's not still getting better. And then if you take the signal away, you get a huge spike. They think they've just run downhill and they try and turn around and reverse it. Does that make sense? So this pathway is really taking a derivative. And there's a lot understood about how this works. In particular, there's these two enzymes, key R and key B, which methylate the receptors and actually change their affinity for ligand in real time. And this mediates perfect adaptation. So whatever concentration of ligand you put them in, they will come back to the same steady state. And very roughly, the key A concentration or the key A activity, how quickly these are phosphorylating, represents E. coli's best estimate of whether they're going uphill or downhill, not of their current concentration. Does this sort of make sense? Okay, so what we want to do is estimate the information rate to key A during gradient climbing. So we want to understand how much information does the key A signal get about d log CDT. Okay, so in the low signal limit, you can actually estimate the information rate and you can write it very compactly in frequency space. So the information rate is this integral over frequency space of the signal strength times the kernel between the signal and the output. So that here is going to be the key A divided by the intrinsic noise in key A. So this is a signal to noise ratio, and this 1 over 4 pi comes out of a more careful calculation. Okay, and to really measure this for E. coli, we need three things. One is K of T. This is the kernel between a step change in concentration and the response of the kinases. That's K of T. We're going to have to measure that. We also need to know the signal statistics, S of omega, or the autocorrelation of the signal that they see. And then we also need to see the noise correlations. Okay, and I'm going to explain how these are all calculated. To do this, I want to tell you about this really remarkable FRET tool that Kata has developed. So this is the microfluidic device. These are not real bacteriums, but the E. coli are glued to the bottom of this cover slip. And what Kata can do is he can change the concentration of ligand that they see basically in real time. And while he's doing that, there's a FRET reporter. And the way that works, the molecule that dephosphorylates QI, so brings it back to its steady state, you can attach one color of fluorophore to that and a different color of fluorophore to QI. And then if they're in close proximity, the spectrum of light that they emit will change. And by doing this, you can see how associated they are in real time. So in the experiment, which you can see here, what Kata does is he alternates increasing the concentration, that's these red bars here, and decreasing the concentration. And as he does this, he can look at individual cells and see how they interpret this signal in real time. And from this, he can infer a linear kernel of how a step change in concentration maps to the dynamics of key A, if that makes sense. These are really individual cells and individual times, but by averaging, you can get a nice smooth curve like this. And this is really well fit by two exponentials. There's a rise time and a decay time. Okay, the other thing we need is the noise spectrum. And to do this, he just puts these same cells in the same setup, just in a constant background, and just looks at their intrinsic fluctuations. There's really a ton you could notice about these experiments. I think they're super interesting. These cells are really each kind of unique. There's a lot to go into here, but the short version is that you can extract an autocorrelation curve of kinase activity, and it's actually pretty well fit with a single exponential. So individual cells have a lot of variation, which maybe is interesting, but it's pretty consistent with just being fit by a single exponential. Okay, and one of the things that actually makes this system really unique is that, and I think is a limitation for a lot of studies of information theory and biology, is that we actually also know the signal statistics. And that's because, unlike in most systems, the signals that they generate or the signals that they see are really just generated by their motion in a gradient. So recall that what we're calling the signal is just d log c dt. And if they're in an exponential gradient with steepness g, the signal then is just the x component of the velocity, right? And so if I want to know the autocorrelation of signal as a function of time, all I need to do is measure their correlations in velocity as a function of time, and multiply it by the gradient steepness squared. Okay, and this.
I'm sorry, but probably I had to ask this. You're assuming that the tumbling motion, the tumbling motion never makes a mistake.
No. So actually, the way these experiments work, they're put in a dish that doesn't have a gradient, and they're just tracked as they swim around in the gradient, or in the lack of gradient. Sorry. And from that, you can measure the autocorrelation of their velocity. So, and the assumption is that in a very shallow gradient, this would generate a very weak signal, right? And that's the signal that they would see. So there's a linearization assumption that their swimming statistics in the absence of gradient would be roughly similar to their swimming statistics in a shallow gradient. Okay, so this is what individual tracks look like. And from these, you can calculate a velocity autocorrelation function. There are some caveats here. Also, this autocorrelation function actually doesn't look very exponential. And one thing that we don't really have the ability to do is know whether a single bacterium's autocorrelation would really be non-exponential, or if the reason the average one is that there are different cells with different tumble biases, so that there is a lot of heterogeneity in the population, and we don't have the ability to test this, but what we assume is that the average cell really would have exponential correlations, and that's what we use. Okay, so I've told you how we measure these three things now, right? And this lets us calculate the information rate that these bacterium would see in a shallow gradient, and the result is that the information rate can be summarized as this constant beta times the gradient squared. The signal statistics have this factor of g squared in them, and in a shallow gradient, we expect this to be the information rate, and that makes a prediction for the information rate as a function of the gradient steepness. Okay, and just to give one more data point here, in a 10 millimeter gradient, that's sort of the shallowest gradient that people do these experiments in, these cells would get about 0.002 of a bit per second. Okay, so finally, I want to ask whether E. coli are information limited. So in the first part of the talk, I was saying that the drift velocity is really bounded by some function of the information rate. And now we've calculated the bound and this information rate, and now we just need to check whether they're close to it. And just to stress this, the bound could be far from saturated, right? So it could be that the Ki signal is really getting a lot of information about the environment, but it's not using this effectively to chemotax. Or it could be that the bound is very close to saturated, right? And just to quantify this, we can talk about the efficiency, which is really how close actual cells are to this bound here. Okay. It turns out that measuring climbing speed in a very shallow gradient is actually quite difficult. There's a lot of details that I am probably poorly equipped to explain about how to generate a very stable, very shallow gradient. And there's also issues about making sure that what you're not seeing is just that if you start all the cells in one area, they'll start to move towards the other area. But at any rate, we could do this, and so there's spread both in the X and Y direction, or I should really say that Henry did this. And from this, you can see that these are all in the shallow gradient regime, and that we can pull out this chemotactic coefficient, which is the linear constant that describes in a shallow gradient the relationship between the climbing speed and the gradient. So now we have these two different functions of gradient steepness. One is the drift speed and one is the information rate. And these two y-axes are the x and y axis of this plot. And so now we can put cells on this. And here's where they are. And this suggests an efficiency of about 65%. So yeah.
So is this the efficiency? I guess I have two questions.
Yeah.
As you just alluded to, is this the efficiency with which the cell uses Ki, or also the efficiency with which Ki observes the gradient? That's one question. The other question is, and forgive me because I think the answer is implicit if I watch the talk again, but are you optimizing just within run and tumble strategies, or is this really within other motility strategies? Like what if they were arranged like little fish? What if they, did smoother changes in direction and so on?
Okay, the second question has an easier answer, which is that this is all in the context of this run and tumble model that we have. So for example, you could ask whether steering would be helpful. Mm-hmm. And there's good reasons to think that that's not feasible for bacteria, sure. But that's not something we can address here, or it's at least it's not something we have addressed here. And I think there's good reason to think that they really don't steer; they just run and they tumble, because I guess for similar reasons of being too small to measure, it's also hard to know whether turning left or right is better. The other question is about what this is the efficiency of. And what I would say is it's given how much information Ki gets, and we don't have a good understanding of how much it could get. I would have more to say about that actually. But given how much it gets, how quickly could it climb the gradient? So for example, it could be that though Ki has a very good measure of whether they're going up or down, this isn't communicated well to the motor, right? And the motor is just tumbling randomly. Right. That's what would be low efficiency. Another thing that would be low efficiency actually is if Ki was getting information, but the information was already out of date. So our information measure includes information about what the gradient or what the what the x component of the velocity was 10 seconds ago, but that's not actionable anymore for chemotaxis. And actually, so this efficiency is a combination of those two things, right? Some of the information might not be good, and some of the information might not be getting to the motor. And we can't really distinguish those. I think that was your first question in its entirety. Awesome. Okay, so just to conclude this part, E. coli need very little information to chemotax, so something like 1/100 of a bit per run, much smaller than the 1 bit that you might think. They also get very little information, so in a shallow gradient, they have this huge apparatus, and it's trying, but it's not getting a lot of information. And then I think this is very different from the usual information theory analysis. So what's often done is calculating how much information a system gets. And I think here what we're trying to do is instead ask how the signaling fidelity really limits the behavioral performance. I don't know if this is getting at all at your earlier question. So this isn't, anyway, maybe.
Yeah, I mean, I would think, to be quite honest, naively, that the reason you're getting an answer that's a factor of 2 off is because the bacteria is actually, to reproduce, is facing a Bayes optimal decision theory problem. And whether that can be approximated with an information theoretic framework or not is another question, but I would almost say it's tautological that that's what it's facing, and that might explain the factor of 2.
Yeah, so as in there could be other things that it's trying to also optimize.
It's introduced,
yeah.
It's ultimately going to be interested in something like reproductive fitness, which is going to have to do with how long it's going to be surviving, how much of whatever it is that's trying to accumulate it can be using to actually reproduce, what the threats are. It's going to have to do with the distribution over environments it's likely to encounter.
So I have one thing to say about this. I mean, maybe 2 things. One is that one thing that's sort of nice about this system is that it does seem that it's really designed to climb gradients. So that seems to be what most of the information is for. And actually, it climbs gradients for many different things. This very simple idea that it's just trying to get to food is probably not its main job.
Okay, we could make the objective function be, anyway, we can talk about this offline.
Yeah.
Whatever the objective function is, yeah.
But maybe another thing that is interesting and speaks to this, they tumble too often, and there's various explanations in the literature about why this is. The one I find the most compelling is that they're actually bred not to chemotax in liquid, but they're put on auger plates. And actually in auger plates, there's this additional problem that if you just swim in a straight line, you get stuck a lot. And the idea is that on auger, but not in solution where these experiments are done, the optimal strategy really is to take more tumbles so that you avoid getting stuck. And I think it's a fair point that they could be optimized for things like that are not in this setup. Yeah.
Well, you only have 10 minutes for part 2, so
I see.
I'm going to ask another question.
Okay, maybe I will go quickly to part 2. Okay, so the second part of the talk, and it will be shorter than the first part of the talk. This is work done by Sam, the PhD student with me who will be graduating soon. Okay, so the big question here is what does biology use its energy for? Some of it goes towards wasted work. I won't show this video again in the interest of time, but for example, E. coli needs to spin this propeller to move itself. And when you hike uphill, you generate a lot of heat, right, which you don't get back when you walk downhill. And this is really in the purview of thermodynamics, right? But I would argue that some of this energy is really going towards information processing, very loosely defined. So I don't mean anything in particular here. except maybe things that don't fall into this category of thermodynamic wasted work. And just for some examples, we spend a lot of energy proofreading molecules, so making sure that DNA is copied accurately into more DNA and RNA and proteins. Also synchronizing molecules, and what I'm going to be talking about today, sending information. And these costs are large, so our brain consumes a lot of our energy. And much of it is actually used just towards pumping ions across the membrane. So you spend a lot of your energy pumping sodium out of your neurons and pumping potassium into your neurons. And the only function that this serves is that it opens a channel for very fast communication. Okay, so information has to be sent over a lot of scales. And this problem that I've been telling you about, information needs to get from the sensory apparatus at the nose to the motors about a micron away, and it needs to get there in about 100 milliseconds. In neurons, information needs to be sent more like over a millimeter, and this has to happen in milliseconds. And one thing you could ask is what's the fundamental cost to sending information? And there's sort of a fundamental cost depending on how you think about it. So copying information is free, but erasing it costs energy. And this would suggest sort of a kBT per bit cost of sending information.
Why is copying free?
I think this is the usual picture of Landauer's, right? That I can copy information for free, but then if I want to erase it, that costs kBT log 2 per bit.
Depends whether by copying you mean overwriting something which might have had a bit before, as opposed
Fair enough.
to,
Yeah, so in many cases, the copying and the erasing is done at once, and then you have to pay. If you're saying that in this example, that would always be true, I would agree.
Okay.
So I would say there's a fundamental cost, which is something like kBT per bit to send information. But what I want to argue is that this is, in any case, very far from the actual cost. And what I want to ask is, there a practical cost that we can understand? Okay, and to explain this, I also want to say that in addition to being sent over a lot of different length and timescales, there's also a lot of diverse physical mechanisms. So for example, there are electrical signals where ion channels mediate currents, which depolarize distant parts of the membrane. And then you have sensors which look for that depolarization. You also have diffusion of second messengers. So QI, the molecule that was a central part before, it gets phosphorylated and then it diffuses to different mechanisms, to different sensors. And then we use sound, right? So we use our vocal cords to generate sound, which travels to your ears, where it's detected by specialized hair cells. And there are some people who claim that cells use this also. And of course, this is not an exhaustive list. But the questions I want to ask are, what determines what strategy cells use when? And what is the minimum energy that cells could use to send information? Okay, how do we understand the order of magnitude of this cost? And the hypothesis is that basically all of these things do the same thing, right? They send information from one place to another place. And the strategy should be chosen to minimize the cost of sending the information. And that here is going to be the energetic cost. And the optimal strategy could depend on how far you want to send the information. It could depend on how fast you want to send the information. And it could depend on how large you're willing to make your sender and your receiver. And just to stress this, these aren't fundamental bounds that I'm going to be talking about. These are bounds that really reflect the reality of biology that takes place in sort of warm, salty water carried out by molecular machines. Okay, so I want to, let's see. If I spend 5 more minutes, is that okay?
Yes,
Or, Okay. Cool. So let me just quickly tell you how electrical signaling works. So the medium through which the signal is sent is that the cytoplasm is a conductor. It's an ionic solution, and the plasma membrane is a capacitor. And you spend an enormous amount of energy pumping sodium out and potassium into the cell. And this accomplishes two things. So first, it maintains a constant voltage gradient across the membrane. So the inside of your cells are negatively charged compared to the outside. But in addition to this voltage gradient, the pumps maintain chemical gradients. And the reason they do this is so that an ion channel can open and send a current in either direction. So if I open a channel to sodium ions, that will depolarize the membranes because there's a lot of sodium outside. If I open a potassium channel, that will hyperpolarize the cell because potassium is at very low concentration outside, and so it will flow out. And these signals are received by mostly voltage-gated ion channels, which sense the local voltage across the membrane. Okay, so the setup we're going to be thinking about, the input is going to be a physical current. In particular, for the case of ion channels, we're going to assume that what an ion channel can do is it can mediate a time-dependent current, which is Gaussian shaped around its origin with some radius sigma. And the receiver is going to be some channel that's some distance away, some distance r away. And what it's going to measure is the charge density as a function of time over a similar Gaussian of size sigma output. So that's the setup. And we're going to be calculating the cost of sending information through this physical channel. Okay, so I'm going to go quickly through the details here. But the things that we're going to need to calculate are the response function. So if I let in a little current at time t here, what's the Green's function of that at the receiver? And I'm also going to need to calculate the noise spectrum at the receiver. I'm going to use the same function for the information rate that I had before. And the other thing that I'm going to need now is a dissipation rate. So depending on the power spectrum of the input, I'm going to need to know how much power that takes. And then what you should notice is that both the information rate and the dissipation rate are integrals over frequency space that both have this input power spectrum in them. And so by dividing the integrand, what I can come up with is a signaling cost, which is really a cost per bit to send a signal through this physical channel from the input to the output. Okay, and to calculate this, I need the transmission coefficient, the dissipation function, and the noise function, the receiver. Okay, so the dynamics for lambda are actually pretty interesting because charge is carried, or the currents are carried through 3 dimensions, but charge can only pile up at the membrane. Maybe I won't attempt to give too much intuition for where this comes from, except that there's 2 physical constants, the conductivity and the capacitance. And once you have these dynamics, you can easily calculate the susceptibility. You can also calculate this dissipation function, and there's many ways to do this, but one is to just calculate the dissipation through the bulk resistor as you send the signal. And then to calculate the noise, we make the very strong and no doubt incorrect assumption that the dominant noise is thermal. There's certainly no getting around that noise. And then with this, we can calculate the noise spectrum using the fluctuation dissipation relations. And the end result of this is a cost in the form of kBt per bit to send the information through this channel. And it has this particular form. So the cost increases as the square of the distance. The cost is also inversely proportional to the size of the sender and the receiver. So a larger sender has less resistance, and so it's easier to send. And a larger sensor can better detect small signals. And And then it's cut off with this universal function of the ratio of the distance you're sending it to L of omega. And I have to explain what that is. So if R is larger than this, the cost rises exponentially. L of omega, it turns out, is alpha over C, and I'm sorry, omega is at the bottom there. But to explain it, you're probably used to thinking of an RC time.
Could you maybe for people?
Yes. Okay, so here alpha over C is an RC speed, and you cannot send a signal faster than this without paying an exponential cost. Okay, so you could also send signals through two-dimensional diffusion and three-dimensional diffusion. I'd be happy to chat more about this after, or through sound, maybe I'll end by telling you about this phase diagram for when you should use these different strategies. So if you want to send things over short distances, you should use diffusion. If it's slow, you should use 2D diffusion. And as you want to send things faster and over longer distances, you should start using electrical and eventually sound. Okay, maybe that's a good place to stop. I'll put my conclusions up here. And I'll stop for questions.
Great. We asked a lot of questions during the talk. People who need to leave, please feel free. But there are a lot of questions asked during the talk. Do we have any other ones?
Are those your students in postdocs on the lower right?
Yes. Mostly.
And so that will be it too. And the other talk again.